It's my great pleasure to introduce Jeff this afternoon. Number one. Jeff did his bachelor's and honours and PhD, all at the University of Melbourne, across the road. He went on to do a postdoc at the NIMR in London with Tony Holder, not Tony Hodder, even though it was also in malaria. Returned to do a postdoc at WeHi with Ray Norton in 2003, and has been a lab head at WeHi since 2010. Jeff's very accomplished, massive number of publications that I was looking at, over 2,500 citations, and that's reflected in the awards that he's received over the years, including every acronym you can think of, an APA, a CDA, and an ARC Future Fellow. His very first publication was on the evolution of thyroxine-binding proteins in native Australian marsupials. So I don't know if you had to collect the blood yourself, but that sounds pretty interesting. And now he's working mostly on the SOCS Jak-Stat pathways that I'm sure you hear about today. But some lesser known facts about J.J. Babon. He's 1/8 Indian. So I feel a kinship that I didn't realize before this morning. He's also read Lord of the Rings more times than you can imagine. And he does cut his own hair.
Who told you all that?
I have my sources. Anyway, it's my great pleasure to introduce Jeff. Please welcome him.
That's the best intro I've ever had. Thanks, Shaolin. All right, so Shaolin asked me to speak about hematopoietic cytokines and their receptors, and that's pretty much what I'm going to do. Obviously, talking about those cytokines, I'm going to mention the signalling pathway that they use. And then at the very end, I'll talk about how that pathway is regulated. The regulation of cytokine signalling is my sort of area of study, but I'm not going to bore you with too much of that. It's only at the very end we'll talk about its regulation. All right, so to start with hematopoietic cytokines, I have to start with the process of hematopoiesis. This will be very obvious to probably many of you, hopefully not all, but I'll go through it anyway. So hematopoiesis is, of course, the process whereby hematopoietic stem cells give rise to all of these mature circulating cells of cell types that you'll all know about: red blood cells, platelets, granulocytes, T cells, B cells, for example. Now, there's relatively few hematopoietic stem cells. They all live in the bone marrow, but there's vast numbers of circulating cells that are turned over regularly, frequently. And so the whole system has to be churning out large numbers of cells every day from just a few hematopoietic stem cells. And so the process is highly efficient, it's highly ordered, because we need the correct numbers of all of these sorts of cells, and it needs to be tightly controlled and regulated as well. And that's what hematopoietic cytokines do. They regulate this process. So as a stem cell becomes a committed progenitor cell, either in the myeloid lineage or the lymphoid lineage, then it has to start making some decisions. It's got to start to proliferate to begin with. It's then got to differentiate and choose which way it's going to differentiate. And then these cells have to make a number of sub-decisions as they go down this differentiation cascade in order to give rise to the final mature circulating cells. And they don't make these decisions on their own. They're driven towards making particular decisions by exposure at the right time of various hematopoietic cytokines. So this is a picture from a paper of one of Lorraine Robb's papers from 2007. On it, she's listed the majority, not all, but the majority of the important hematopoietic cytokines, and most of these I'll be talking about today. So what are they? GCSF is granulocyte colony stimulating factor. As its name suggests, it drives formation of granulocytes. It was discovered here at WeHi by Don Metcalf a few decades ago, and it's an incredibly important therapeutic as well as being an important hematopoietic cytokine. GM-CSF drives formations of granulocytes and macrophages, and again, this was discovered here at WeHi by Nick Nicola. MCSF drives formation of macrophages and monocytes. Stem cell factor is important in stem cell renewal. EPO and TPO, you've no doubt heard of, drive formation of red blood cells and platelets. And there's a number of members of the interleukin family that we consider as hematopoietic cytokines as well. In particular, members of the IL-2 family are important in T and B cell development. That's interleukin 2, 4, 7, 9, 15 and 21. And there's a couple that aren't in this picture that I'll also talk about today as well. LIF is leukemia inhibitory factor. That was discovered again here at WeHi by Doug Hilton, and it's a member of the IL-6 family of cytokines, which I'll also touch on. So knockout of individual cytokines in mice have been quite informative in terms of what they do and how they function. In general, knocking out a cytokine leads to a relatively severe phenotype with hematopoietic abnormalities in the mouse. Rather sort of unexpectedly, it's only 2 of them that are lethal. So if you knock out stem cell factor or EPO, the mice die of severe anemia whilst in utero because they're effectively making no red blood cells. The other knockouts are in general tolerated to lesser or greater degrees, but they do give rise, as I said, to hematopoietic abnormalities. And in the case of knocking out the IL-2 family of cytokines, you can get a strong immune phenotype as well, because of course you lose large numbers of T cells and B cells. So as part of putting this talk together, I thought I'd go through and try to find all of the cytokines, the hematopoietic cytokines from this diagram that are in the clinic as FDA-approved therapeutics. I was reasonably surprised that there's not that many of them. In terms of hematopoietic cytokines, of which there's a relatively large number, there's relatively few that are used. There's just this sort of 7 or 8 here. So G-CSF is used to treat neutropenia, which is low neutrophil numbers. You can see from this diagram that makes sense. G-CSF in humans has this interesting side effect that it mobilises the stem cells to come out of the bone marrow and into the circulating system. And so it's also used for stem cell harvesting in patients that need that because it avoids going into the bone marrow. EPO is, of course, used to treat anemia. TPO is used to treat low platelet numbers. Sometimes these are used to treat athletes that want to run faster. Growth hormone is used to treat short stature. Leptin is used to treat leptin deficiency. Interleukin 2 is used in a couple of late-stage proliferative diseases, neuroblastoma and renal cell carcinoma, but that's pretty much about it. There's a handful of anti-cytokine antibodies that are also in the clinic, and they're in general used to treat a variety of immune and autoimmune and inflammatory diseases. So the purpose of a cytokine, which is this blue sphere here, is to act as an intercellular messenger and to give an instruction to a target cell. And it's going to do that by binding to a specific receptor on the surface of its target cell. There's going to be 1 receptor per cytokine. Each cytokine is going to use a different receptor. And it's going to give an instruction to that cell, and that instruction may be to proliferate if we need more of them, to differentiate in terms of being exposed to progenitor cells, to cause them to differentiate down one of those blood cell lineages. It may tell the cell to apoptose if there's too many of them, or it may just tell the cell to do something different, like secrete certain factors or express certain receptors, for example. Just do something it wasn't doing beforehand. So really you can think of it as a soluble intercellular messenger. Now, like all signalling pathways, the way that it's going to work is that it's somehow going to send a signal into the nucleus, which is going to tell certain genes to switch on and be transcribed. And like most signalling pathways in multicellular organisms, that's going to be driven by phosphotyrosine-based signalling. So phosphotyrosine-based signalling cascades are a hallmark of all multicellular organisms. They're only found in multicellular organisms, apart from the single-celled organisms that have retroevolved back from being multicellular. And they all use the same key components. There's a tyrosine kinase, an enzyme called a tyrosine kinase, which will write the signal by phosphorylating the tyrosine on a protein. That signal, that tyrosine phosphate, can be read by proteins that contain SH2 or PTB domains, so they're capable of binding to phosphotyrosine residues. And the signal can also be erased by classes of enzymes called tyrosine phosphatases that get rid of the phosphate and get the protein back to its unphosphorylated form. And so as I go through the pathway that hematopoietic cytokines use to signal, these sort of three key players will keep coming up. All right, so cytokines signal through cytokine receptors, that's obvious, but the nomenclature here gets a little bit tricky because really they're divided into two classes, one of which is also called cytokine receptors, which isn't particularly helpful. The other class are called receptor tyrosine kinases. So most of the cytokines that I showed in that first slide signal through cytokine receptors, but stem cell factor and MCSF signal through what are called receptor tyrosine kinases. Now, as their name suggests, the thing about these receptors is that they have tyrosine kinase domains encoded in their intracellular regions. So outside the cell is the ligand binding domain, then we have a transmembrane domain, inside the cell will be a tyrosine kinase domain. And that's what's going to start signaling. So the cytokine is going to bind to the receptor. That's going to stimulate that tyrosine kinase domain to start working and phosphorylate things, and that's what initiates signaling. Now, cytokine receptors work in the same way, but because they don't have their own tyrosine kinase domains, they have to recruit a family of kinases, and they're called the JAKs, or Janus kinases. And once again, they'll be in charge of initiating signaling by phosphorylating something on a tyrosine. So it's mostly because, for two reasons. One, that's the area I know best, and two, most hematopoietic cytokines use this pathway. It's this pathway that I'm going to concentrate on in today's talk, and I'll just be mentioning this in passing a couple of times. All right, so in a bit more detail, in a schematic form, here we have a cytokine. It's a small soluble secreted factor. It's going to bind to a receptor on the surface of its target cell, as I said before, and it's going to initiate signaling by getting these Jak molecules to become activated. So JAKs, like most tyrosine kinases, are present in an inactive state under normal conditions. Tyrosine kinase signaling is almost always proliferative, and so it's very dangerous for them to be switched on and active when they're not supposed to be. So most tyrosine kinases are made in a switched off form, and they're activated by some subsequent signal. And the signal here is the cytokine binding to its receptor. Now, the receptor is always going to have at least two chains, sometimes more, and two of those chains will be bound to a Jak. So the way that these JAKs are activated is that through a process that we don't fully understand, cytokine binding to the receptor induces them to activate one another in trans by phosphorylating each other. And when they do that, they're active, and then they do what tyrosine kinases do, which is to phosphorylate tyrosines on protein substrates. Now, again, one of the hallmark of tyrosine kinases, as opposed to serine threonine kinases, is that they're relatively inefficient enzymes. So they only tend to phosphorylate things that they're directly bound to. And this is true with the JAKs as well. So here you can see that the first things that they phosphorylate are the receptors to which they're attached. And then these newly created phosphotyrosine sites can be read by a family of transcription factors called the STATs. The STATs have an SH2 domain, and that's what enables them to bind to these phosphotyrosines. And then once they do that, they're in turn activated again by phosphorylation. They go into the nucleus, and they drive the biological response by upregulating the transcription of the appropriate cytokine-inducible genes. Now, at the same time that they're doing that, the pathway is going to need to be switched off at some point so that the cell can return to its basal state. And so what the STATs are doing at the same time is that they're upregulating the transcription of their own negative feedback inhibitors, the SOCS proteins, which I'll go into later. These are going to feed back in on that signaling cascade and switch it off, forming a negative feedback loop. So it turns out that the Jak-Stat SOCS signaling system is pretty well conserved throughout evolution. It's present in organisms from insects onwards, as is the case for many signaling pathways. It's become more complicated in mammals compared to more ancient organisms like Drosophila. So, for example, Drosophila has only 3 cytokines, 1 cytokine receptor, 1 Jak called hopscotch, 1 STAT, and 3 SOCS proteins. Humans, on the other hand, have over 40 different cytokines, an equivalent number of receptors, 4 JAKs, 7 STATs, and 8 SOCS proteins. So looking at where they arose in evolution, you probably can't read a lot of this. This is an evolutionary tree going from bacteria, eukaryotes, plants, all the way to mammals. So we first see cytokines and cytokine receptors appearing at the onset of bilateral symmetry. So insects are a good modern-day equivalent of that. But these cytokines and cytokine receptors use pathways that they built up from previously existing components. So the SOCS and Jak molecules were already there at this stage of evolution. They're found in animals with radial symmetry. The STATs are the oldest of all. They're found in plants as well as animals, suggesting that they arose really at the dawn of eukaryotic evolution. So we're not just interested in JAKs and STATs because a lot of the Institute works on cytokines and the immune system. We're interested in it because it is one of the key signalling pathways that controls mammalian, and in fact all vertebrate development. So if you're like me, you go to a lot of talks, there's a bewildering array of signalling pathways and signalling molecules that people talk about, and it seems incredibly complicated, but in fact there's only 8 main classes of signalling pathways that use cell surface receptors to drive development of the organism. This is TGF-beta, TNF-alpha, cytokines themselves, receptor tyrosine kinases, notch, Wnt, hedgehog, I've forgotten one, and G protein coupled receptors. So they all have certain things in common, and that is the fact that they use an extracellular ligand to bind to a specific receptor, and then they're going to translate a signal into the nucleus in the form of an activated transcription factor. But how this signal gets from the surface of the cell into the nucleus differs dramatically between these different pathways. For some of them, like the Jak-Stat or cytokine signalling pathway, it's relatively simple. In others, it's incredibly complicated, like TNF signalling. If you've been to any of John Silk's talks, is a very complicated way of getting a signal from the outside of the cell into the nucleus. Receptor tyrosine kinases often act through a wave of activation of enzymes. So one kinase will activate another kinase, which will then activate another kinase, and so on, before you finally end up with a transcription factor inside the nucleus. Some of these pathways seem ridiculous from an engineering point of view. So Wnt, for example, the transcription factor involved in Wnt signalling is beta-catenin. Beta-catenin is always being made and degraded inside your cells, and it only stops being degraded when the cell receives a Wnt signal. So you're using all of this energy to make and degrade a protein that you're probably never going to use or only need to use very infrequently, which seems like a crazy design. Probably the most elegant signalling system is Notch. With Notch, the receptor itself is also the transcription factor. So when Notch binds to a ligand, the receptor's cut in half. The bottom half goes into the nucleus and it acts as the transcription factor. But apart from Notch signalling, Jak-Stat is the most direct. So we have the cytokine, we have the receptor activates one kinase, the kinase activates one transcription factor. So a few of this, you would have seen this before. I have a sporting analogy I like to use to explain how elegantly efficient this process is. This is one of the best AFL goals you'll ever see by the Hawks a few years ago. We have one player kicking it in, Guerra. It's marked by a second player. He handballs it to Buddy, the third player, who kicks one of the greatest goals you'll ever see from about 80 metres out. So 3 players getting the signal from one end of the cell, if you like, to the other. Whereas if we want to have a look at TNF signalling or RTK signalling, it's a lot more complicated. The signal is transferred from one player to another. Almost every player in the Geelong side is used to get this goal. There's something amazing about this, but it's not exactly what you'd call efficient. We're getting there. So you'd think it'd be here? No, one more handball. There we go. Okay, so what I'm going to do for the rest of the talk is to go through the components of the cytokine signalling pathway one by one. So we'll start with cytokine, we'll go to receptor, we'll go to Jak, we'll go to STAT, and we'll end up with its negative regulation in terms of the SOCS proteins. All right, so cytokines. So the cytokines that signal through the Jak-Stat pathway and through the so-called cytokine receptors aren't all hematopoietic cytokines, but a lot of them are. But there's also a number of inflammatory cytokines as well, such as the interferons that you've probably all heard about. Now, historically, they've been divided into two classes, type 1 and type 2. Most of the hematopoietic cytokines are type 1 cytokines. In some ways, this classification is a bit artificial. It's just based on sequence motifs in the receptors that they use. But really, structurally, all of these cytokines are very similar. They all arose from the same common ancestor, so that's not surprising. So as well as the hematopoietic cytokines that I've talked about, we've also got the interferons and a few members of the interleukin family, 10, 20, 22, 28. So here's some structures of a handful of cytokines: LIF, EPO, GCSF, interferon alpha and gamma. They're all essentially the same structure, apart from interferon gamma, which is a dimer of the same structure, but they're all 4 helix bundles. And those helices always have a certain topology in terms of which ones are up and which ones are down. That 4 helix topology is really just there to provide a scaffold for various variable loop regions to be different between each cytokine, and it's those variable loop regions that will generate specificity for one receptor and not another. So they're going to use these variable regions to bind to the receptor. The receptors, unlike the cytokines, have a large variety of different shapes and overall structures, but they do have some things in common. So this is an example of the LIF receptor. As I said, there's always at least 2 chains. The LIF receptor consists of 2 chains, one of which is called GP130 in blue, one of which is called the LIF receptor beta in red. Now they're well structured outside the cell, and that's the part that's going to bind to the cytokine, shown here in green. Then they have a transmembrane domain, but inside the cell, they're completely unstructured. So most cytokine receptors will have anywhere from 300 to 600 residues of unstructured polypeptide inside the cell, and that's what I'm representing by these dotted lines here. So those regions are responsible for recruiting other signalling components. So at the top of this region right next to the cell membrane, we have the areas that will be responsible for recruiting the Jak-Kinase, and then below where the Jak-Kinase sits is really, in a lot of ways, unimportant. It's just there to provide tyrosines at certain positions. Those tyrosines are going to be phosphorylated by Jak. They're then going to be used to recruit the STAT proteins, and that's what's going to start signalling off. So it's really there just to place certain numbers of tyrosines in the right position. Many of them will also have a negative regulatory tyrosine at some point, and that's the tyrosine that's going to bind to a SOCS protein, which is going to end up switching off signalling. Okay, so they're often, this is now a schematic diagram, obviously, these aren't real structures. That's a very obvious thing to say. This slide is to indicate that people, and myself included, often lump all of these cytokine receptors into various groups. So on the left, we have the cytokine families that use shared receptors. So for example, the IL-3 family of cytokines, 3, 5, and GM-CSF, they each have their own dimeric receptor that consists of 2 different chains in each case, but one of those chains is always the same. So for example, IL-3 uses a receptor with an IL-3 specific alpha chain, but the second chain is called the common beta chain, and that's used by the other members of that family as well, which will have their own specific alpha chains, but they'll have this beta chain in common. IL-2 family cytokines generally signal through trimeric receptors, and again, one of these chains is going to be shared by the receptor that every member of that family uses. That's called the common gamma chain. The IL-6 family of cytokines is the 3rd class of shared receptor, and all of the IL-6 family cytokines that are listed here use a receptor that has at least 1 copy of the protein GP130 that I showed you just in the last slide. As well as those, we have EPO, TPO, leptin, growth hormone, and GCSF receptors. They're the ones that are homodimers, so 2 copies of the same protein chain form up those receptors. They're different in each case, so 2 EPO receptor chains form the EPO receptor, 2 TPO receptor chains form the TPO receptor, and so on. All of the others, IL-12, 23, 413 and the interferons, they're all heterodimeric receptors, so copy each of two different chains, make up those receptors. And in general, the way that the cytokines uses the receptor to signal can be in one of two ways. The simplest and the most intuitive is that you have two receptor chains that aren't joined together in the absence of cytokine. They're floating around in the cell membrane. It takes a cytokine to come and cross-link those two receptor chains. That brings the JAKs close together, and that initiates signaling because they're able to activate and switch each other on at that point. However, probably most, at least half of the cytokines that we know about bind to receptors that are already preformed on the surface of the cell. And the belief here is that they're in a conformation that doesn't allow the JAKs to activate one another, and it takes a cytokine binding to that receptor in order to induce a conformational change that brings the JAKs together and once again allows them to signal. So here's some structures of some real cytokine receptors. So this is the EPO receptor. It's a homodimer of two chains. It's cross-linked here by EPO, shown in green. That allows the two chains to come close together, which allows their associated JAKs to come close together, and that's how signaling occurs. So that's the most simple system, a small homodimeric receptor. GCSF is relatively similar. It also signals through a homodimeric receptor, so the two chains shown here in red, but this time it takes two molecules of cytokine to cross-link the two chains with the same end result, that's going to bring JAKs close together and start signaling. IL-2 family cytokines, like I said before, use a trimeric receptor. Here the situation is a bit different. So in the back here we have the IL-2 receptor alpha chain. It's tethered to the membrane, but it doesn't bind to a JAK. Okay? It has effectively no intracellular domain. Its purpose is to grab hold of the cytokine, and then it's that cytokine receptor complex that is recognised by the other signaling chains of the receptor. So it's the blue and the red. The blue is the common gamma chain. The red is the IL-2 receptor beta chain. It's only those two that are bound to a JAK, and once again, they're going to initiate signaling. So it turns out that this mode of action is used by a lot of cytokines. There'll be a specific alpha chain which does not bind to a JAK, but which is there to recruit the cytokine, and then it's that cytokine receptor complex that's able to cross-link the two signaling chains, which are bound to a JAK inside. This is IL-6 family cytokine signaling. As I said before, there's a range of different stoichiometries in terms of the receptors that they use. So the simplest system is LIF, for example, shown here in green. LIF basically just cross-links a heterodimeric receptor, one chain of GP130, one chain of the beta receptor. CNTF is similar, but it needs an alpha chain in order to do this. So the alpha chain binds to CNTF, and that complex is then able to cross-link GP130 and the LIF receptor beta. IL-6 uses a hexameric receptor, so it takes two copies of cytokine, two copies again of an alpha chain that does not bind JAK, and two copies of GP130 in order to get signaling to occur. The most complicated system is GM-CSF, and the IL-3 family cytokines. They're believed to signal through a dodecameric complex, so 12 proteins, 4 copies of cytokine, 4 copies of a non-signaling alpha receptor, again in light brown, and although it's not obvious here, there's 4 copies of this blue beta common chain, and all of that is required to get the two chains in the middle, which are the JAK binding chains, in order to activate JAKs and initiate signaling. All right, so speaking of JAKs, we'll move on to them now. So the JAKs were discovered again here in Melbourne by Andrew Wilkes when he was working for Tony Burgess in the Ludwig Institute. He used the newly invented technique of PCR in order to go searching for novel kinases. And he found a couple of kinases that had a very different domain architecture than any of the others. And he called these the JAKs. Now, he started off by calling them the JAKs, he tells me, because it stood for just another kinase, because it wasn't what he was looking for. He then, when he found this, they had this certain domain architecture, He sort of thought that name and decided that it stood for Janus kinases, because Janus is the Roman god of beginnings and endings, has two heads, and JAKs are the only kinases that have two kinase domains, or one of the only kinases that have two kinase domains. So there's 4 domains in a JAK. There's this firm SH2 domain, which binds them to the receptor. The catalytic domain is this red one, the kinase domain. In between is the characteristic feature. It's another kinase domain, which is catalytically inactive. It's called a pseudokinase domain. Structurally, it's a kinase domain, but it's lost its catalytic residues. And in some way that we don't understand, it's there to regulate the activity of the kinase domain. So it turns out that there's 4 JAKs encoded in all vertebrates: JAK1, JAK2, JAK3, and TYK2. And knockout of those JAKs has shown how important that they are and has really placed them at the head of the cytokine signalling cascade. So knocking out JAK1 and JAK2 is lethal. Knocking out JAK3 gives mice severe combined immunodeficiency with very low T and B cell numbers, suggesting that this JAK might be involved in IL-2 signalling, because that's what generates T and B cell numbers, and that turns out to be the case. TYK2 knockout mice deficient in, amongst other things, interferon alpha signalling, and TYK2 loss of function mutations in humans give rise to an immune disease called hyper-IgE syndrome. JAK1 knockout mice die shortly after birth because they don't respond to a range of different cytokines. JAK2 knockout mice give the most interesting phenotype. They basically don't make any red blood cells, so they die early on in embryogenesis. And that, of course, suggests that JAK2 is somehow going to be involved in EPO signalling. And so it's studies like this that have enabled us to build up a picture of which JAKs are responsible for which cytokine signalling pathways, so they can't all act in all pathways. There's a lot of specificity in the system. So, for example, interleukin-2 family cytokines always use a pairing of JAK1 with JAK3. One of them is going to activate the other, or they're both going to activate the other. JAK3 is always attached to the common gamma chain of the receptor, whilst JAK1 is always attached to the cytokine-specific alpha chain. IL-6 family cytokines always use a pairing of JAK1 and JAK1, although there is actually some redundancy in that system, unlike most of the others. IL-3 family cytokines, EPO, TPO, growth hormone, always use a pairing of JAK2 and JAK2, and so on and so forth. So there's no point memorising all of this. The key points to remember are that JAK3 is the most specific. It's only used by IL-2 family cytokines. TYK2 is the second most restricted in its use, but JAK1 and JAK2 are very widely used. And it's also only JAK1 and JAK2 that ever act in pairs, so a JAK1 with a JAK1 or a JAK2 with a JAK2. The other, you never get a JAK3 pairing, for example. All right, so those JAKs are then going to phosphorylate and activate some STATs, heading further down the pathway. STATs are transcription factors, which means that they're DNA binding proteins. There's been some really beautiful structural work giving us structures of an activated STAT in this case. As I told you before, they have an SH2 domain, which is shown in brown or orange or whatever that is. So that allows them to bind to the cytokine receptors that have just newly been phosphorylated. And it is also what allows them to dimerize in this particular orientation, which poises them to wrap around DNA, which is shown in this structure here, and use their DNA via their DNA binding domain. And it's that, of course, that enables them to then act as transcription factors. So once again, knockout of STATs have been very informative in telling us what pathways that they're involved in, and once again, there's specificity here in terms of STATs, so not all STATs can be used by all cytokines. So I guess a little bit surprisingly, it's only STAT3 knockout that's embryonically lethal. But knockouts of the other STATs show impairments in particular signalling pathways. And this has allowed the following picture to be developed. So in the same way that specific cytokines use specific receptors and those receptors recruit specific JAKs, the receptors also recruit specific STATs. So for example, growth hormone only ever uses STAT5, IL-6 mostly uses STAT3 with a bit of STAT1 thrown in there as well. Interferon gamma mostly uses STAT1, and so on and so forth. All right, so that's the stats. So now, obviously, signalling is going to have to be switched off at some point, and it's been known for a long time that overactive Jak-Stat signalling is seen in a wide variety of cancers. It's obviously a very proliferative pathway, and therefore it has the potential to be cancerogenic, if that's a word. And so it had been clear to everybody that it's very important in cancer. However, it's only been relatively recently that mutations in components of the Jak-Stat pathway have been directly shown to cause cancer. The first really came about 10 years ago, when it was found that 3 what are called myeloproliferative neoplasms were caused by a single point mutation in Jak2. These 3 diseases, polycythemia vera is an overproduction of red blood cells, ET is an overproduction of platelets, myelofibrosis is a more severe disease where, amongst other things, you get scarring of the bone marrow. But anyway, most patients with those 3 diseases have a particular point mutation in Jak2. It's always a somatic mutation rather than a germline mutation. There's been, I'm about to say not many, I don't know of any germline Jak or Stat mutants off the top of my head. So all of these are somatic mutations. So since the discovery of that particular point mutation, it's really been the cancer genomics studies of the last 3 to 5 years that have shown that there's in fact mutations in a number of Jak and Stat components that can give rise to a variety of different leukemias, lymphomas, and even in some cases solid cancers. And the one thing that all of these mutations have in common is that they're always activating. So they lead to either Jak or Stat being constitutively active, even in the absence of cytokines. So these pathways are switched on when they shouldn't be. The other way that Jak is found to be mutated in disease is by oncogenic fusion. So here you have a translocation that ends up fusing the kinase domain of a Jak with a chunk of protein X, whatever that is, and depending on what protein X is, that can also give rise, in relatively rare cases, to a number of different leukemias. So as I said before, all phosphotyrosine-based signalling pathways use phosphatases to control the signal, and there's some very important phosphatases that regulate Jak-Stat signalling as well. So Jak is activating itself and phosphorylating receptors and stats, and then phosphatases come along and dephosphorylate it, trying to send the system back into its basal state. But at least in terms of Jak-Stat signalling, it seems that it requires an extra level of regulation where it uses feedback inhibition to make sure that the pathway gets back to it, or the cell gets back to its unstimulated state as quickly as possible. And that's the function of the SOCS proteins. So the SOCS proteins, unlike the phosphatases, aren't constitutively expressed. So the cell makes large numbers of them when signalling is active in order to turn it off at the right time. So the SOCS proteins were discovered, again here at WEHI, by Doug Hilton and Robin Starr in the mid-90s. They used an assay that now many of us are using again to try to discover things like this, which is based around the ability of a cell line called M1 cells, which can form nice colonies in agar under normal conditions, but you expose them to interleukin-6. They differentiate into a macrophage-like cell, and they form these dispersed colonies. So he realised you could use this as a phenotypic screen. If you took a cDNA overexpression library and you used it to transfect a culture of M1 cells, then what you look for are colonies that grow even in the presence of IL-6, because that means that they will have incorporated a cDNA that has switched off IL-6 signalling. So that's what they did. They discovered this clone 4A2, and when he sequenced the cDNA inside it, it encoded a protein that he went on to call SOCS1, or suppressor of cytokine signalling 1. Now, as I told you earlier, there's 8 SOCS proteins in humans, SOCS1 through 7 and cis, and they all have the same domain architecture. And here's our SH2 domain popping up again, as it does in all tyrosine-based signalling pathways. So it's going to bind to a tyrosine phosphorylated protein of some sort. And the SOCS box, it turns out, is able to recruit the cell's proteasomal degradation machinery. So the way that SOCS proteins work in general is that their SH2 domain will grab a phosphorylated signaling molecule, and the SOCS box will recruit the components needed to degrade that signaling molecule. And we have a pretty good idea of how this works now. So here I've drawn this SOCS protein like a little fish. The tail is the SOCS box, and the fin is the SH2 domain. So its tail grabs a protein called a longan BC. The fin, the SH2 domain, grabs the phosphorylated substrate. When that happens, it can dock on to a protein called cullen-5, which is the scaffold of what's called an E3 ubiquitin ligase. Probably a lot of you know what E3 ubiquitin ligases are. They're enzymes that can transfer ubiquitin to, in this case, a substrate. So along with the rest of the ubiquitination cascade, the activating enzyme, the conjugating enzyme, the end result is that ubiquitin is transferred to the substrate. Polyubiquitin chains are formed, and that polyubiquitinated proteins with this topology are recognised by the proteasome and degraded. And so it's all a way of getting this substrate ubiquitinated and then degraded. So the SOCS proteins are what's called the substrate recruitment modules for these E3 ubiquitin ligases. One half of the SOCS protein interacts with the ligase, the other half grabs the substrate, and then the substrate is degraded. We have real structures of these sorts of complexes. This is a SOCS2 based E3 ligase, SOCS2 in green, everything else as it's coloured here. And if we model in where a substrate would bind, it sits on one end of this scaffold, activated ubiquitin would bind here, get transferred to the substrate gets polyubiquitinated and then degraded. You can do these experiments in vitro if you have the patience. If you make all of these components, put them into a test tube together and add ATP, you can see that over time the substrate the substrate band is disappearing, and you're getting this ladder of higher molecular weight products which represent mono and di and tri ubiquitinated species. So by and large, the SOCS family works this way. It grabs hold of an activated cytokine receptor. So these tyrosines as part of the intracellular subunits of the receptors that aren't bound by STATs but are instead bound by SOCS proteins. The SOCS proteins grab hold of that and they cause the whole receptor complex to be ubiquitinated and degraded. So switching off signalling by getting rid of the activated receptor. And the consequence of this is that the SOCS proteins, because they're a negative feedback inhibitor, they don't affect the amplitude of the wave of signalling, but they affect the duration with which signalling stays on. So this is shown here. Here we have wild-type cells on the left, SOCS1 knockout cells on the right. You can see if we expose those cells to interferon gamma, signalling comes on after about 15 minutes. Signalling is measured by phosphostat-1, and then after a certain time it's switched off. But in the absence of SOCS1, you can see there's an extension in the time that signalling is still active. In SOCS2, the same is true in response to growth hormone. Again, at later time points, you still have signalling going on, and SOCS3 knockout cells in response to IL-6. Now, even though this is at an admittedly subtle molecular phenotype, there's usually just, if you measure it, a twofold increase in the length of time that signalling is active for if you remove one of these SOCS proteins. It still leads to drastic consequences for the organism. So SOCS1 knockout mice die as neonates from a lethal inflammatory disease that's driven by this level of excess interferon gamma signalling. SOCS2 knockout mice are giants because of this level of dysregulated growth hormone signalling. SOCS3 knockout mice die in utero because of this level of dysregulated LIF signalling, LIF being an IL-6 family cytokine. So to put that schematically, the purpose of the SOCS proteins, if this is a wave of signalling going on, is to decrease the half-life of that wave of signalling, but not really to affect its amplitude. And that's how most of the SOCS proteins work. The only exceptions to that are SOCS1 and 3. They can do that, but they don't do it very well. We now consider them a separate class of SOCS proteins. They're the two most potent inhibitors of signaling, particularly inflammatory signaling. They're the only two that are active in the absence of their SOCS box. So if you get rid of their ability to ubiquitinate anything, they can still control signalling. And that's because they have what's called a kinase inhibitory region at their N-terminus. And this allows them not just to bind to receptor, but also to bind to Jak at the same time and directly inhibit Jak's catalytic activity. So this is a much faster way of switching off signalling than having to recruit all of the ubiquitination and proteolytic components that the cell has. And I guess it's also more reversible as well because you're not degrading the whole receptor complex. We now understand how that process works. This is a structure of SOCS3 in green, bound to Jak2 in light brown, the active domain. And the kinase inhibitory region is this tail at the N-terminus of SOCS3. And that tail, as you'll see in a second, is sitting in a groove on the surface of its Jak target in here. Now that groove is usually where, if we model in where a substrate would bind in white, you can see that groove is usually where the substrate would bind. So it's not completely blocking the substrate binding site, but it's blocking it well enough to stop any substrates getting in. If Jak can't bind to a substrate, clearly it can't phosphorylate anything, so it's to all intents and purposes inactive. And that's how SOCS3 and SOCS1 are able to inhibit signalling so efficiently. This method of inhibition isn't just used by cytokine signalling pathways, also by at least one other, which is receptor tyrosine kinase signalling. So this is the insulin receptor kinase, shown in brown. The insulin signalling has its own negative feedback inhibitor, so like a SOCS protein, called GRB14, and it works in the same way. It puts its kinase inhibitory region, which is called the BPS, in the substrate binding groove, and it switches off its target kinase, the insulin receptor kinase, in the same way. So SOCS1 and 3 can use this mode of inhibition to inhibit Jak1, Jak2 and TYK2, but not Jak3, because Jak3 is missing a key motif that all of the other JAKs have that allows SOCS1 and 3 to grab hold of it. And we don't really understand why, but there's obviously some benefit to the organism in having Jak3 uninhibitable by SOCS. SOCS1 works in the same way, it's just 10 times better at it. So it inhibits its target kinases, the same three, 1, 2 and TYK, with 10-fold lower KDs, which means 10-fold better affinity. So now we can add SOCS to our sort of Jak-Stat cytokine pathway and look at which SOCSs take care of which cytokines. And again, there's a lot of specificity in the system. So SOCS1 and cis take care of IL-2 family cytokines. SOCS3 takes care of IL-6 family cytokines, GCSF and leptin. SOCS1 takes care of IL-12, 23, 4, 13 and the interferons. Growth hormone is taken care of by SOCS2. So the real gaps in our knowledge here is that we don't know if there's any SOCS proteins that are capable of inhibiting IL-3 family cytokines, EPO or TPO, and they're very important hematopoietic cytokines, and we're very interested in trying to find out whether they are inhibited by another member of the SOCS family or another member of a different protein family, or whether they just don't require this kind of regulation, which seems unlikely. So that's pretty much it. This is just a schematic which was published a few years ago, which probably explains the whole system. We have hematopoietic cytokines. They bind to receptors that activate JAKs. The JAKs activate STATs, and that leads to upregulation of specific genes and gives the cells an instruction such as differentiate. It's regulated both by phosphatases at every level and also by the SOCS proteins, which can directly inhibit them and also cause their proteolytic degradation. I'd just like to leave you with one sort of caveat slide. So it turns out that a number of different cytokines, as well as inducing STAT-based signalling, also co-induce other pathways at the same time. And LIF is a good example of this. LIF induces the phosphorylation of STAT3, which leads to activation of the STAT pathway. But at the same time, it's also co-activating or co-stimulating both the MAP kinase and PI3 kinase pathways as well. And in the case of LIF, these actually have opposing functions. So when you use LIF to signal to ES cells, The STAT3 is trying to keep them self-renewing, whilst the active MAP kinase pathway is trying to make them differentiate, and the PI3 kinase pathway is trying to get them to survive. So it's often the balance between which of these three pathways are most active that leads to the end phenotype. So whilst it's true that most cytokines are dominated by STAT-based signalling once JAK is switched on, there are, in certain cases, other pathways that are activated at the same time and are also driven off the same receptor. So I just wanted to sort of fess up that I'd maybe oversimplified a lot of it. But anyway, that's my last slide, and thanks for listening.
Thanks, Jeff. I think the audience will agree with me. That was a really clear, informative talk, one of the best we've had. So thanks, thanks very much. Questions?
How does the phosphorylation STAT association with the
That's a good question. Do we know that? So certainly the phosphorylation of STAT leads to, and in fact I should have put a structure of the inactive and active, so the unphosphorylated and phosphorylated forms. They're like scissors. The ones that you saw look like that, but the ones in the unphosphorylated form look like that. So whether that conformational change is what's getting it off the receptor or not, to be honest, I'm not 100% sure, but it probably is. Yeah.
Question down here first.
Do not happen to if it's functional if you swap the tails between the cytoplasmic tails between cytokine receptors?
Yeah, I've been trying to get an honest student to do that for the last couple of years because I think it'd be really interesting. So you should be able to do that, and you could make, for example, if you did it in the TPO, if you put a SOCS3 site in the TPO receptor. So the one thing I didn't say is that all of the cytokines end up in expressing almost all of the SOCS proteins, but they are specific for certain pathways because there's not the right sites in the receptors. So if you put a SOCS3 binding site in the TPO receptor, TPO will initiate the transcription of SOCS3, and by our model, it would then go and inhibit it. But we've never done it, unfortunately, but it's a really good idea. Maybe you'd like to do it.
Yeah, recruitment drive.
Okay.
Lynn.
Mine's a question of stoichiometry, because when you look at a Western that has phosphostat and stat on it, you find the phosphostat changes with kinetics, but that the constant pool of unphosphorylated stats up. Is it stats something or
Probably stats, yeah.
well, anyway, so it must be a small portion of the total that's phosphorylated of the total amount of protein. And also, I wonder about that if you're in the proteasomal degradation, or I might not. It's the SOCS box I'm thinking of.
Okay.
And so that has an affinity for the elongins, but it won't go to, won't be prepared for proteasomal digestion unless there's a phosphorylated protein bound there. So I wonder if they can block up the system by having an abundance of non-phosphorylated over phosphorylated.
The stat you're talking about, of stat forms.
I guess I'm talking about the stats.
Yeah. So actually, sometimes when you do Western blots and the stat antibody won't recognise the phosphoform, which there is a couple of antibodies that are like that, you see that in some cases you are getting, in fact, the majority of stat being phosphorylated. But you're right, in most cases it's a small proportion of it. But the SOCS proteins aren't recognising the stats; they're recognising the receptors. And so it's the receptors that they're going to grab hold of and cause to be ubiquitinated.
Matt.
How important is the role of phosphatases in the whole system? I think you must like SHP2 phosphatase. Yeah. Like in the absence of a particular phosphatase, is the whole system completely dysregulated?
Not really. So I did look into this at some point, not personally, but I read all the literature based around that. So in general, you see a much milder effect than you do in the absence of a SOCS protein. So if you knock out individual phosphatases, you see that the cells become slightly hyper responsive to cytokine, but it's not crazy. It's maybe by 5-fold or something. So it's nowhere near as severe a phenotype as if you get rid of a SOCS protein in terms of the regulation. What tends to happen is that it's the amplitude of the signal is affected, but because the SOCS proteins are still there, it's switched off at the right time. Whether anybody's done a phosphatase and a SOCS knockout on top of one another, I'm not sure, but that would be interesting because that should really stuff things up, you would think. Yeah.
David.
So if the SOCS proteins ubiquitinate the receptors,
Yep.
how do the receptors get to the proteasome? Is there a vesicle that comes in, or are they plucked out of the membrane?
I've asked every proteasome expert that I've ever met at a conference that question. So the best answer that I got is that there's proteasomes everywhere, and they'll be taken up to the receptor. They're found everywhere. Yeah, I don't know if that's true or not, but that's what he said. Yeah.
Just over here.
Thanks. You spoke of the role of Jak activating mutations in some cancers. I was just curious how a parasite may be able to upregulate stats and yet avoid that kind of effect on the host cells and the host at large.
Is that what happens? Is it parasites can?
They can avoid they avoid the proliferation of the cell as well. And that kind of seems counterintuitive, really, from listening to your talk.
Well, I guess a parasite for a while is quite happy with as much cell proliferation as possible. No, because it ends up getting more cells if it can go along with it. But you're saying it's completely stopped, is it? I don't know. That's interesting. I'd never thought of that before.
So there's no reason, what else does the STAT pathway do?
Well, it's not always causing proliferation, I guess. In the examples that I gave of diseases it is, but often, well, I guess mostly it's causing proliferation, but yeah, not always. I don't know. That's really interesting. I never thought of that before.
I might ask one last question, Jeff. So you've given a nice diagram of the combinatorics of cytokines down to the signaling pathways. In terms of the transcription factors or other genes that they're transcribing, is there a relationship there with any one of the components of that signaling pathway, or is it semi-random, or what does that look like if you overlay it on?
The only one that I really know anything about is STAT1 and 3, so they're very different. The gene sets that they upregulate are basically mutually exclusive, and in fact you get, in humans now they've found that a number of STAT1 gain of function mutations work like STAT3 loss of function mutations and vice versa, so they really appear to be working at against one another. But as for the others, I'm not sure. Yeah.
Let's end it there. Please thank Jeff for an excellent seminar.